team at Invitae. On behalf of everyone here at Invitae, welcome to our webinar, Stand By for Launch, an in-depth look at the data from an RNA analysis pilot study. First, I'm going to start things off by quickly running through a few housekeeping items. So first, all participants are muted. We will have a Q&A session at the end, and you can submit questions at any point along the way during the presentation using the questions pane of your webinar control. I also want to take a moment just to acknowledge the questions that were submitted during the registration process. We will do our best to address as many questions as possible. Prior to this role, she held positions as a staff scientist in the applications group at 10x Genomics and was the clinical genomics application manager at Personalis. Our second speaker is Dr. Keith Nijkamp. He holds a doctorate in molecular genetics from the University of Florida and was a postdoctoral fellow with the Howard Hughes Medical Institute at the University of Wisconsin-Madison. Dr. Nijkamp leads the clinical sciences and interpretation team here at Invitae. We're delighted to have both Dr. Garcia and Dr. Nijkamp speaking today about the fundamentals of RNA analysis as well as presenting data from a pilot study. And then our last slide before moving into the presentation are the disclosures for both speakers, Dr. Garcia and Dr. Nijkamp. And so with that, we'll begin the presentation by turning things over to Dr. Garcia. Welcome, Dr. Garcia.
Thank you so much for that introduction. I'm always genuinely excited when I'm asked to present on the wonderful world of genetic testing and how our field and the industry is pushing to make those tests better and better to provide more answers to our patients. I'm going to walk you through some of the fundamentals today of RNA and RNA analysis and the challenges and opportunities that this new tool provides us with. So because this is a talk about RNA for genetics professionals, I'm pretty sure it would be illegal for me not to have a slide on the central dogma. So here it is. But in all seriousness, I am going to come back to this figure over and over today, because while we know that these three molecules are critical in the functioning of the cell, we also tend to ignore whatever molecules we aren't assaying at the time. So in clinical testing, usually we're trying to correlate information that we can gather about DNA to explain or predict a phenotype. But of course, that line between the assayed molecules and the phenotype is anything but a direct, straight line. And so today, the focus of my section will really be on all of the complexities that make this a very circuitous line to phenotype, especially as it relates to RNA. One advantage that DNA has in all of this from an assay perspective is that it's a lot simpler than RNA. It's really stable. It's relatively constant from cell to cell. There's usually only 2 copies in any cell, and it tends to hang out in the nucleus. So this means that for most germline applications, you can get your DNA material from any source, blood, skin, biopsy, any tissue you have available, and you don't expect the results to differ by that source. But on those axes, RNA is a whole lot more complex. The number of copies of RNA varies per cell quite drastically, but on average, mammalian cells have somewhere on the order of several 100,000 mRNA molecules. RNA is also processed and transported, so RNA in different stages of maturation coexist within the same cell. And there are many different types of RNA, so that you have to purify out the ones you're interested in. But one of the advantages is that in that central dogma, RNA is one step closer to the phenotype. But we have to keep in mind the complexities and the variability of RNA from cell to cell in order to properly interpret the results from RNA assays. I'm not going to spend a whole lot of time today talking about how RNA assays work, because for the most part, if you know how an NGS-based DNA assay works, then you're 90% there for RNA. Because once we've isolated the RNA, our first step is actually to just turn it right back around into DNA. And we call this new molecule cDNA or complementary DNA. It looks just like DNA, A's and T's, C's and G's, and so it can be sequenced just like DNA. The only real difference is that it's missing a few things, namely the intronic sequences and any exons that have been spliced out. So it's representative of the mRNA, but looks like DNA. So why are we so interested in assessing splicing anyway? Well, when we see a change, a DNA change in clinical sequencing, central to what we're doing when we're interpreting that variant is trying to predict how is that variant going to disrupt the cell and then ultimately the person. Is it a missense variant, a synonymous variant? And there I did it. We've kind of gotten into the habit of jumping right to the protein. We tend to assume that RNA won't change beyond that one variant change, but we have to recognize that that is an assumption. There are lots of other paths to wreaking havoc on the cell that aren't so direct from DNA change to expected amino acid change. And splicing is a great example of how the actual impact of a DNA change can be much, much greater than just translating the 3-letter codon into an expected amino acid change. But when we talk about splicing variants and splicing signals, our minds usually go to the 2 bases at the intron-exon junction. This is sometimes called the canonical splice site, the essential splice site, the donor and acceptor dinucleotides, or the +1, -1, +2, -2 splice positions. And you'll probably hear me go back and forth between all of those at some point during this presentation. And these bases are critical in the biochemical reaction that actually results in the cutting and rejoining of the DNA that results in splicing. And variants at these positions are generally considered to be likely pathogenic just by virtue of their position, because these are very highly conserved bases, and we know that they're critical to that fundamental reaction of splicing. But let's be honest here, there's a part of all of us that are interested in genetics because it's so complicated. So, of course, the story is more complex than this. There are actually many signals in the DNA and resulting RNA that help to direct the splicing process. Instead of walking through a whole lot of that detail today that you're very unlikely to remember, what I really want you to take away is that the canonical positions at that intron-exon junction are just one of the signals that are needed for splicing to occur. But there is significant conservation of sequence around those bases. You can see that I have these plots at the bottom for the donor and the acceptor sites, which are ways for us to look at what does conservation look like around a splice site. If you take all known splice sites and stack them all up and ask how likely is it that each base will be at each of these positions, what immediately stands out to you are those dinucleotide GT and AG positions. But you can see that immediately outside of that, there is a preference for which bases are present just outside of those dinucleotides. We can see that there's a polynucleotide tract that's immediately upstream of the acceptor. And there's also a critical A base that we call the branch site. And that can slide around a bit relative to the acceptor site, and thus it's harder to predict. There are also various splice enhancing and splice silencing signals represented by little green and red boxes in this figure. And those recruit different proteins to the splice region and direct how splicing will actually occur in any given cell. And variants at any of these locations, in the exons or in the introns of the DNA sequence, may potentially disrupt splicing and lead to disease. So how do we know whether a variant is likely to impact splicing? Well, luckily for us, there are algorithms that are designed to do just that. Very similar to SIFT and polyphen that we use to predict the impact of missense variants, we also have splice prediction programs that help to predict which DNA variants might impact splicing and the degree to which that variant might disrupt the splicing pattern. But also like missense algorithms, they each have different strengths and weaknesses, and they take different things into account. Some may only predict the effect of a variant at a canonical splice site. Others may be able to predict the gain of a new splice site. Others may be able to predict splicing enhancers or splicing silencers. But if we use these algorithms on clinical samples, we can take a look at where these variants tend to live. So here, this is based on an analysis of 689,000 patient samples that were processed at Invitae who underwent genetic testing for a range of hereditary disorders across lots of clinical areas. And what we see is actually most of the predicted splicing variants are within the exonic sequence. And that's at least in part due to an ascertainment bias, in that most of these patients were assessed using a panel where the primary purpose is to sequence exonic sequence and only a little bit into the intron. But what we also see is that there's a huge enrichment of pathogenic and likely pathogenic variants at that 1-2 canonical dinucleotide sites. And this is, of course, in line with the ACMG recommendations and what I already said we know about the importance of these positions. But if we get outside of that 1-2 dinucleotide positions, then you're seeing a huge number of variants of uncertain significance. Because just like SIFT and polyphen, if we're relying on in silico predictors to predict the effect of the variant, those aren't robust enough to change the classification of a variant. And so we end up with a lot of variants of uncertain significance in these predicted potentially splice impacting regions. This also reminds us to really check our assumptions about how strong of a correlation there is between variant type and disease mechanism. We tend to assume that missense variants cause disease by changing an amino acid, leading to protein dysfunction in some way. Similarly, we tend to assume that synonymous variants don't cause disease because the amino acid doesn't change. But in fact, if these exonic sequence variants are predicted to potentially affect splicing, then it could be that these relatively minor changes to DNA actually result in quite catastrophic protein changes like frame shifts and protein degradation. And this is not theoretical. There are plenty of examples of synonymous variants, for example, in LMNA, that cause a frame shift and protein degradation because of their impact on splicing. So we have to keep in mind that there are many ways a variant could impact cellular function. We also have to remember that our assays are not fully representative of our patients. Whenever we're interpreting in vitro assays, we have to remember that deleterious does not mean pathogenic. Just because we can demonstrate a functional defect in vitro, just because we can observe a splicing defect, this does not mean that that defect is enough to cause a phenotype. Not every variant that causes a discernible splicing defect will ultimately be disease-causing. And the opposite is also true. Just because you see no change in splicing, this does not mean that you have demonstrated that the variant is benign. And part of that has to do with the whole sample type complexity and RNA complexity that I referred to earlier. Not all tissues express or splice genes in the same way. Here's just one experiment that demonstrates that. Here we performed whole transcriptome analysis, so sequencing of all of the cDNA that was isolated from the RNA of the cells, on a set of blood samples looking at the expression of 84 genes that comprise a multi-cancer panel. And we found that only 16 genes in that set of 84 had enough coverage to meaningfully be able to assess them. Here we defined that as needing to have seen at least 10 transcripts per million reads sequenced. But that doesn't leave us with nothing to do next. In very much the same way that we increase coverage on our DNA assays by reducing the footprint, we can do the same in RNA. We can improve our situation by targeting the sequencing to only the 84 genes that we're interested in. This is basically like going from a genome or an exome to a panel in order to increase coverage. And when you do that, you can recover most of the genes, but not necessarily all of them. In this sample experiment, there remain around 8 genes that aren't expressed at a high enough level in blood to be able to make meaningful conclusions about them on an RNA analysis. And I'm not trying to make any statement about these particular genes. It's just important for us to note that not every gene that we're interested in from a clinical testing perspective will be able to be assessed from RNA analysis if you're using blood as your sample type. And even if your gene of interest is expressed in blood, that doesn't mean that the splicing pattern in blood is the same as the splicing pattern in the relevant disease tissue. There are cell-to-cell differences that drive different cells to splice transcripts in different ways. Here I'm just showing a visualization of the alpha-tropomyosin gene on top, and the at least 6 different splicing patterns that are at least partially specific to particular tissues. So let's say I had a variant that was predicted to affect splicing of the green exon. Then I may very well need a smooth muscle biopsy in order to be able to see whether that variant impacted that splicing. Because that green exon is not present in other tissue types, if I looked at striated muscle, I would see no difference from a control population in my patient cells because the green exon is never expressed there. And that brings to light another current limitation of RNA analysis. In any RNA assay, you're going to be using a set of controls that you're going to use to help you assess whether you see a difference in splicing pattern. And you also, when you're designing these assays, need to understand whether blood is a good representative tissue to use, and you'd like to be able to compare the splicing patterns you see in blood to the tissues that you might be interested in from a disease perspective. But in order to know any of that information, we really need to know a lot about RNA expression in different tissues and in different individuals, so that any differences we see, we can place in the context of how likely is this to really be different versus just a stochastic change between our patient that is not meaningful for phenotype. But frankly, we're just at a much earlier stage in our understanding of RNA. The GTEx portal is one place that we tend to go to look for information about RNA as compared to GNOMAD, which is our new gold standard for comparing differences in DNA. And what you can see is we just don't have the same kind of data on RNA that we do on DNA. And this isn't new for us in genetics. In fact, it's incredibly analogous to what our lives looked like before we had resources like GNOMAD. We're just at an earlier stage in our understanding of RNA variability between tissues and between individuals. And so we have to recognize that we run the risk right now of overestimating the pathogenicity of RNA changes because we don't yet have a great understanding of what that background level of variation looks like. So I want to take a few minutes now to talk about how we interpret splicing variants with and without RNA assays and what the guidelines currently say about these topics. I'm going to walk through a few examples here, and we're going to need to agree on a few key assumptions. That the variant in question is quite rare. It's in a gene where we know loss of function variants are the mechanism for disease. And that if the variant does impact splicing, it does so in one of those catastrophic kind of frame shift loss of protein ways. We also need to assume that we know absolutely nothing else about these variants except their position relative to a splice site. They're completely novel. So if the only thing we've done is DNA sequencing, and we find a variant at the splice donor or acceptor site, as I said before, we assume that the variant is likely pathogenic. This is true of the ACMG variant interpretation guidelines and the variant interpretation framework Sherlock that we use at Invitae and that's published in Genetics and Medicine. So how does this change if we had RNA data? Well, most of the time we expect that the RNA assay for these donor and acceptor variants will show a splicing defect and support the likely pathogenic classification. Although just having that information about the RNA assay isn't necessarily enough to push it all the way to pathogenic. The more likely classification change that will take place is a downgrade to a VUS if the RNA assay happens to show no change in splicing. Now, if we move to a position outside of the canonical splice site, so this is we're less sure that this variant will impact splicing. Our only indication that it may impact splicing is from one of these predictors like SIFT and polyphen. On the missense side, we have a prediction from one of our splice algorithms saying this one might impact splicing. Well, very similar to the situation where we have a missense variant and the only thing we know is that SIFT or polyphen predicts that it might be deleterious. If we have that kind of information on a potential splice variant, we still are going to put that variant at a VUS classification without any additional information. But again, what happens if we can use these variants in an RNA assay? Well, again, if we have just this piece of in silico information on top of the prediction that this is a splice site variant, that's not necessarily enough information to change that variant's classification from a VUS up. it is potentially enough information for us to downgrade that variant to likely benign, but we're still in one of these sort of uncertain categories. And part of the challenge of interpreting the kind of RNA splicing data that we have available is that the RNA data is actually pretty complex itself, and Keith will show us some nice examples of that. One single DNA variant can cause one or more RNA effects. You can skip an exon completely. You might include part of an exon or an intron. There are lots of different things that we can see in an RNA assay. And I tend to think of this as the one DNA variant to multiple RNA effects as being the cell kind of struggling to figure out what to do once it's lost its splice site. So just for example, let's say that we took out the splice acceptor in exon 2. Well, the splice donor in exon 1 still needs to go somewhere. It's trying to splice to anything it can find. And it's kind of scrambling to figure out a new place that it can use as an acceptor now that exon 2 is gone. And that means that sometimes it might find exon 3, sometimes it might find the middle of an intron, sometimes it might find the middle of exon 2. And so you can get this spreading of multiple effects when there's actually only one DNA variant present. So it's not the case that when we see a shift in RNA splicing data that it's always exclusively from one species to a different species. You can get many species that emerge. And each RNA effect can have a different impact on the protein. So again, if we go back to that central dogma, just knowing that there's a defect in the RNA is not enough for us to say this variant definitely causes disease. We have to go through the exercise of figuring out, well, with this splice isoform, what will the protein actually look like? Skipping an entire exon might lead to a frame shift, or it may retain the frame and just essentially be a multi-amino acid deletion. Frame shifts early in the transcript will tend to lead to nonsense-mediated decay, and so you don't actually see a altered protein. What you'll see is a loss of protein expression from that allele and only the normal expression from the unaltered allele. Late frame shifts may actually result in the generation of a truncated protein. All of these need to be predicted from the splicing patterns we see, and we have to make a determination about whether those species we may or may not observe are likely to impact the cellular functioning and lead to disease. And finally, these DNA variants can alter splicing to different degrees. It isn't always the case that we shift from one species to an all new set of new species. You can have what we call these kind of leaky splice variants where you get some normal splicing happening from the altered allele and some abnormal splicing happening as well. So I've spent a long time now talking about the use of RNA analysis to aid in the classification of variants. And the use case here is really you have a VUS that you've identified by DNA sequencing, and you're looking for the RNA analysis to support or refute the hypothesis that the variant causes disease by changing splicing. But the second use is also to use RNA analysis in actual variant identification. And this is a case where DNA sequencing might have been negative, and now you're using RNA sequencing to see if you can find evidence for abnormal splicing, in which case you would then need to go back to the DNA to identify the variant that led to the abnormal splicing. So examples of this might be deep intronic variants or perhaps a synonymous variant that was not previously reported on the DNA result. So how often can we expect RNA analysis to clarify or give a new result? Well, most DNA-based clinical tests are negative, but we don't expect to find deep intronic variants in most of these cases. The yield is going to vary quite a bit from gene to gene and from disease to disease. But right now the literature supports that at least in breast cancer patients, we're expecting about a yield of 0.5%. On the other hand, about 30% of clinical DNA results will be uncertain, but only 5% of results overall will have a VUS that's predicted to potentially affect splicing. And then finally, about 2% of all clinical test reports are likely pathogenic or pathogenic with a variant that is predicted to impact splicing. But again, the yield here is expected to primarily be downgrades in classification if any classification change occurs at all. But we can also look to the literature to try and get an understanding of what is starting to emerge about RNA analysis and yield. Most of the conversation around RNA, at least clinically, has been in the oncology space. But there is evidence in the literature that RNA analysis will be broadly applicable across clinical areas. So this is just one publication in which there were 100 cases from the Undiagnosed Diseases Network. So these are cases that are already in the difficult to diagnose category and may not be representative of a general genetic testing population. And the experimental setup was complex. So not all individuals had all assays done, but in this study, they were using exome analysis, genome analysis, and RNA-seq analysis on subsets of patients. But overall in this group, 7 of the 38 diagnoses that were able to be made were made possible by RNA analysis. And of these, 3 were variant classification changes, 2 of which were synonymous variants, and 4 were new deep intronic variants. We can look at other clinical areas as well, but we have to really put these in the context of how these experiments were designed. And these were not experiments that were designed to be representative of a general genetic testing population. For example, in the green box, they looked at 27 families with a child with Fanconi anemia, but in all 27 of those families, the complementation group was identified. So there was already a clinical diagnosis. Some of these children had one variant identified in a Fanconi anemia gene, and the second variant was missing. The same is true in the second blue box of undiagnosed monogenic muscle disorders. This was a patient population that was enriched for patients that had a single variant in a recessive gene or that had a DNA result showing a variant that was predicted to impact splicing. And so you can see that you can get quite a few pickups by RNA sequencing if you have an enriched patient population. And I think that in general what we can say is that the utility of RNA analysis will be increased when the prior probability of disease is high, like the case of clinical diagnosed disease or a single variant detected in a recessive condition. And so oncology may not actually be the highest yield place for the use of RNA analysis. So with that, I just want to wrap up what I talked about today. There are many signals in the DNA that coordinate the process of splicing. We can make predictions about where those signals are and how a variant in those positions might impact splicing. Making those predictions at the canonical splice site is pretty easy, but those predictions are not as accurate as we need them to be. And the fundamental output of this process is a mature processed splice RNA. And so we can go to the RNA to assess the hypothesis that a variant impacts splicing. Though we must acknowledge that by going to RNA, we're diving into a more complex pool of molecules, and we need to understand that we're at an earlier stage of knowing what the background variation of RNA looks like in the population and between tissues. And so you're fundamentally considering multiple hypotheses about how a variant might cause disease, and we can't immediately go to a pathogenic or benign classification on the back of RNA analysis. With that, I'm very happy to hand over to my colleague, Dr. Keith Nijkamp.
Thank you, Dr. Garcia, for a wonderful introduction and overview of RNA splicing and its role in variant interpretation. For the remaining time, I'd like to share with you all our experience with RNA analysis at Invitae from an IRB-approved pilot study analysis or study initiated in 2019. Next slide, please. At the outset of this study, we had 4 major aims. First, we wanted to identify patients